Hello, it's Brett again, and I am here with your Module 4 lecture on cells. So let's go ahead and get into the slideshow. Big. All right. So yes, Module 4, we are looking at cell structure and function. And this is kind of the first part of our investigation of cells. So we are going to be looking at some of the structures inside of a cell. How do these biological molecules that we learned about in Module 3, how do they come together to form even bigger structures with new and interesting properties? And also, this is maybe the first module where we're talking about things that are fully alive. So this image here, this is an image of some special cells called HeLa cells or HeLa cells that are known for, they're a human cell line that is known for their ability to divide essentially without limit. And they were the first human cell line discovered to have this property. There's a really interesting and also very sad story that kind of ties into the history of racism and paternalism in medicine. And I wish I had time for that story now, but I don't. But if you want to learn more, there's a little link here in this file, so you can dig through that and get to that if you are interested. Okay, so what are we going to be talking about in this module? Module 4, I've really got three big ideas I want to try and get across. The first one is cell theory. The next is the general features that are common to all cells. And then the last one is eukaryotic cell structures, and in particular, trying to compare plant and animal cells. All right. So cell theory just has three main statements that are all hopefully not too controversial. The first is that all living organisms consist of one or more cells. So we said before that the cell is the smallest unit of life. Well, this is really saying that living things are made of cells. Okay, sure. Oh, and number two, cells are the smallest unit of life. So even multicellular organisms, you could take cells out of that multicellular organism, and if the conditions are right, they might survive on their own for a while. And there are plenty of organisms that are unicellular, so they just have one, one cell is the entire organism, or perhaps they live in colonies, but each cell in that colony does not really function differently than its neighbors. So, and then the last part of this is that all cells come from division of another cell. So this is kind of an interesting one. Cell division is responsible for reproduction, and there's a couple different types of cell division that we aren't going to talk about in Biology 101. If you happen to know what they are, excellent, excellent. And if you want to learn more, you can ask me, but I'm not going to say those words and potentially confuse people even more, because this module does have a lot of terminology. But I put a little star here because we know this was wrong at least once, right? If we talk to the physicists, they'll tell us about the Big Bang and that this universe hasn't always been here, at least not in the form that it currently is. And so life had to start somewhere. And that first cell on Earth, we think, based on evolutionary theory, we think that gave rise to all of the other life that we see on this planet now over billions of years. But that first cell did not come from another cell. And there are some proponents of, well, maybe it came from an alien planet. Okay, interesting idea. But then that doesn't get away from the problem of where that alien planet's first cell came from, right? So, so we know this was not totally true, but as we see it today, all the cells we know about, they just don't arise spontaneously. They have to come from another cell, or maybe a combination of cells for organisms that reproduce sexually. Okay, so that's cell theory. Now we're going to get on to the general features of cells. How big are cells? Well, the short answer is usually pretty small. So here's a diagram of some size scales. This is a logarithmic scale. So the smallest things are maybe 1/10 of a nanometer, like an atom. Molecules might be on the order of nanometers. Biological molecules might be, 5 to 10 nanometers if they're big biological molecules. Viruses are maybe 100 nanometers. Bacteria are about 1 micrometer. And same goes for mitochondria. And then animal cells and plant cells, these bigger complicated cells, are maybe 10 to 100 micrometers on average. And then if we're talking about eggs, those can actually get to be pretty big. Maybe you're starting about 0.5 millimeter and going up to maybe 100 millimeters, which is pretty big. So very few cells are visible with the naked eye. Most cells we need a light microscope to see. And if we really want to get detailed images of cells, we use a tool called an electron microscope. So there is a neat interactive scale simulator. I'm just going to click on this to open it, but if you're really interested in exploring this, you can use it yourself and zoom in. So there's this, some everyday items, some, like fonts and coffee beans and rice and sesame seeds. And this scale bar up here shows you like 1 millimeter. And then as you zoom in, that millimeters, well, you can't even see the whole millimeter, right? And then you can zoom into smaller and smaller items. So this is kind of like that picture I showed you, but it's pretty neat in terms of seeing the sizes of some of these things that we're talking about. And yeah, we'll get down to a carbon atom. And that PM is picometers, which is really, really small. That's smaller than nano, like 1,000 times smaller than nano. Or I guess a carbon atom would be 0.34 nanometers. We want to do our metric conversion. Anyway, you can play around with that more. I just wanted to kind of point that out. And we'll get back to the slideshow. Okay, well, why are cells small? But I should say, why are they usually small? And the whole idea here is surface area to volume ratio. So as you increase the size of a cube, if you double the side of a cube, the surface area grows as the square, but the volume grows as the cube of that. So for a 1 centimeter cell, for example, the surface area to volume ratio would be like 6 to 1, centimeters squared to centimeters cube, right? And if you double the size of that cell, so if you have a cell that's 2 centimeters on each side, then the surface area to volume is 3 to 1. You increase it to a 3 centimeter cell, it's 2 to 1. And this pattern kind of holds. So the bigger the cell gets, the smaller the surface area to volume ratio is. And what this means is materials have a really hard time getting in and out of the cell quickly enough for the cell to stay alive. So some of the exceptions that we saw on that last slide, like those eggs, those are mostly storage. And then as they divide, it's kind of a very unequal division. So just one tiny part of it becomes the new cells that are going to be metabolizing and growing. The rest of it kind of stays as storage. And then the embryo kind of takes over the inside of the cell and starts to have more efficient transport mechanisms. And that's why those eggs can be fairly big for organisms that lay eggs and kind of just incubate them like a chicken or an ostrich. Okay, some other exceptions. We actually have some cells in our bodies that might be up to 1 meter or maybe 2 meters long. And these would be nerve cells and muscle cells. And they get around this by being really long, but really, really skinny. So, so the cross-sectional area is very small. So the surface area to volume ratio stays very high. We can get materials in and out, but communicating along the length of that cell is sometimes a little tricky. So, I should say muscle cells usually have multiple nuclei and kind of multiple divisions, even though they're one continuous cell. There's usually multiple control centers, so they're kind of a little bit weird. Okay, talked about the exceptions. Okay, well, what are the general features of the cells? What are the parts of a cell? And essentially, there are two main parts. There's the plasma membrane. There's the barrier at the outside of the cell, or the bag, if you will. And then there's the inside of the cell, the contents of the bag, which we call cytoplasm. And that is a technical term for the jelly-like substance inside the cell called the cytosol, and also anything else that's embedded in it except the nucleus. So the cytoplasm you can think of as the filling. The nucleus is kind of special, so it's technically not part of the cytoplasm. And if there isn't an organelle, such as the nucleus, or maybe there's other organelles, these are like little bags within bags that perform specialized functions. Those organelles, except the nucleus, would be part of the cytoplasm, but then the cytosol doesn't include any of the other organelles either. So, and I'm actually going to put this word on here just in case you come across it in our readings. So cytosol plus organelles excluding, actually maybe I'll put an equals here. Nope. Excluding. Yes. Okay. So, so that's the general arrangement of the cell, and yeah. So that's kind of the general features. Clear these before I go to the next slide. Just to preview Module 5 a little bit. We'll take a closer look at the plasma membrane. The plasma membrane is actually really, really interesting because it is this barrier that needs to let certain things in and out and other things it needs to keep out. And it needs to keep the water inside the cell separate from the environment outside of the cell. So to do that, there is a bunch of lipids, and they're actually specialized lipids called phospholipids, which make up most of the plasma membrane. And then embedded in that plasma membrane are several different proteins, and sometimes there's glycolipids, which are used for cell recognition, or glycoproteins, which are used for cell recognition. So there's a lot of cool stuff happening in the plasma membrane. And there's actually 2 layers of these phospholipids, and basically they're kind of like soap molecules that are stacked tail to tail. So there's a hydrophilic part, these little red heads that can interact with either the cytosol or the extracellular environment. And then there's a hydrophobic part, which are the fatty acid tails of these phospholipids. And those prevent water from sneaking through. So it's really kind of a clever arrangement, and we'll talk about it more in Module 5. All right, so those are the general features of cells. And now we're going to get into talking about kind of the more specifically, I'm going to change the color here, more specifically the different types of cells. And there's two main types of cells to think about it. These are prokaryotic cells and eukaryotic cells. Okay, so the prokaryotic cells are smaller and simpler. Prokaryotic cells do not have any organelles, so there's no nucleus, and there's no other compartments within that cell. So these are small and simple cells, and essentially they are the bacteria and the archaea. So there's actually two different domains of life that are both prokaryotic domains. And you can see here, there's just a barrier and then some stuff inside. There's DNA in there. There's ribosomes. There's cytosol or cytoplasm. It's the same thing for these prokaryotes. And that's about it. For the more complicated cells, we call them eukaryotic cells. These are the ones that are bigger. They're more complicated. They have a nucleus in most most eukaryotic cells. And they also have several other organelles in most eukaryotic cells. And we'll talk about those different organelles in a bit. There's only one domain of life that is eukaryotic, and that is the domain eukarya. Oh, that's kind of handy, right? Wait a minute, what do these words even mean? Well, the letters EU is short for, that means true, and karyon means kernel or nucleus. So eukaryotic means true nucleus, right? So they really do have a nucleus. Prokaryote means like before the nucleus. So there's no true nucleus. They have this nucleoid region where the DNA lives, but they don't have a true nucleus. Okay. Well, what kingdoms are we talking about then? For eukaryotic kingdoms, well, there's probably several kingdoms that we used to call a single kingdom called Protista, or Protoctista if you go back far enough. But really this is maybe like anywhere between 10 and 20 kingdoms, I'm going to guess, but they haven't quite figured it out yet. Looking at the genetics, there's just a lot of genetic variation in here. And then those are unicellular eukaryotes. So just single-celled eukaryotic cells. They used to just, that was enough to call them their own kingdom, but now we looked at their DNA and it's like, oh no, if we're calling fungi and plants and animals all different kingdoms, then we need to call Protista many more kingdoms. All right, so then fungi, plants, and animals are the multicellular kingdoms of eukaryotes. And these are organisms that hopefully you are more familiar with. Okay. All right, so prokaryotic cell structure is very, very simple. Let's look at the start. Where's the barrier, right? Well, actually this green layer right here is the cell membrane or plasma membrane. So that's technically the outside of the cell, or that separates the outside and inside of the cell, right? There is some cytoplasm, kind of the light blue just inside of that. There's this nucleoid region where the chromosome lives, and prokaryotes only have a single circular chromosome, or at least bacteria do. I'm not quite sure about the archaea, but I think they have circular chromosomes as well. And then maybe there's some ribosomes in that cytoplasm. That's about it for the inside of the cell. Then outside of the cell, they have another structure called the cell wall, which is very rigid and prevents them from bursting open. And then there's also a capsule surrounding that cell wall, which is like another protective layer. Then sometimes they have these little pili, which are extensions of the plasma membrane, which kind of go poke through the cell wall and the capsule so that the cell can take in nutrients from outside of the cell, or secrete digestive enzymes to dissolve things in the environment, to break down larger molecules. So, and then there's also this flagellum in a lot of prokaryotic cells, which allows for movement. So that's about it. What about eukaryotic cells? Ooh, these are a lot more complicated, right? I'm not even going to read off all of the parts here, but I will say that, yeah, there's still the plasma membrane, right, surrounding the whole thing. There's still cytoplasm. There's the nucleus, which has some separate parts, and then a bunch of other stuff, right? All these other organelles that are doing different things and working together, but kind of each doing their own thing. It's like this orchestra inside, right? And the prokaryotic cells are kind of like the one-person band, right? The singer-songwriter. And the eukaryotes are the kind of the full band with a lot of different parts that are working together. Okay, so there's a lot of new terms here, and a lot of them are really complicated and really long. There's this jumble of terms. How do we make sense of them? Well, there's a lot of different ways we could do this. We could go through it alphabetically, but I like to break these terms up into maybe 3 different groups, and that's kind of how I'm going to present them. The first group of eukaryotic cell structures, these are the organelles with double membranes and DNA, and maybe had an endosymbiotic origin, which I'll talk about in a little bit. These are the nucleus, the mitochondria, and the chloroplasts. And then, so that's one grouping. Then there is the endomembrane system, which consists of the nuclear envelope and also kind of the nucleus. So the nucleus kind of shows up in a couple of these, so I'm sorry. But, and then there's the rough endoplasmic reticulum, or rough ER for short. And then there's the smooth endoplasmic reticulum, or smooth ER for short. And then we have some vesicles and also vacuoles. There's the Golgi apparatus and lysosomes. And those are kind of all working together as kind of part of the same system. And we'll talk about that system and how it works together a little bit later. And then we have other structures, peroxisomes, ribosomes, cytoskeleton, cell wall, extracellular matrix. And we could probably, depending on how technical we get, we could have a pretty long list. But we're going to try and keep it not too technical. Okay, so we're going to start out with the nucleus. So this is a vision of our typical eukaryotic cell. The nucleus is the big structure, the big organelle in the middle that contains most of the cell's DNA. And we would call this the nuclear DNA, or the genomic DNA. So this has the instructions for the cell. It's kind of the control center for the cell. The DNA is essentially just instructions on how and when to build proteins. And then those instructions can get copied and sent to other parts of the cell for those proteins to be made. So the nucleus is the control center, and you'll notice that it has a covering. It's encapsulated in this membrane, and it's actually a double bilayer membrane. So there's actually 4 layers of phospholipids. So each one of those, or each pair of them, would be a phospholipid bilayer. So the nuclear, and we have a special name for the membrane surrounding the nucleus. It's called the nuclear envelope. or nuclear membrane. And it has these pores in it, which are called nuclear pores, which are really big to allow the instructions, the copies of those instructions to be transmitted, but they aren't big enough for the DNA to leave. So that's the nucleus. All right, so nucleus, double membranes, and DNA. Okay, there's actually a couple other organelles that have double membranes and DNA. Ooh, that's a weird one. Okay, so I think I might have mentioned mitochondria. Mitochondria are kind of the powerhouses of the cell, or you could think of them as maybe kind of like a coal-fired power plant or an oil-fired power plant. They basically burn things, right? They combine hydrocarbons with oxygen to produce water, and in turn they release a lot of energy. Most of the energy of a eukaryotic cell comes from mitochondria. And mitochondria are kind of weird because they also contain DNA. Mitochondria actually divide inside of the cell. So you have multiple mitochondria that all have a copy of the DNA. It's kind of almost like they're these little aerobic bacteria that are alive inside of a bigger cell, which is kind of weird. Okay. And there's multiple, there's an outer membrane and an inner membrane. That inner membrane is highly folded, has a lot of surface area, and there's lots of enzymes embedded in that membrane. And we'll talk about the purpose of those enzymes in, I think, module 7 is where we'll hit that. So just to kind of preview and foreshadow our discussion of aerobic cellular respiration. Okay, the other double membrane organelle with its own DNA is the chloroplast. And actually there's kind of 3 layers of membranes in a chloroplast. There's an outer one, an inner one, and then inside of that, there are these thylakoid membranes, which have all of the pigments which can absorb light and help with photosynthesis. So chloroplasts are the organelles responsible for photosynthesis. We color them green because plants are green, and the reason plants are green is because chloroplasts are green, because they have these pigments that absorb kind of everything except green light. Okay, but again, we're getting ahead of ourselves. We'll talk about that a lot more when we talk about photosynthesis. So there are other types of plastids, which are little storage compartments, but the chloroplast is a special one because it's got its own DNA. And again, it's kind of like this photosynthetic bacterium that is alive inside of a plant cell, and those chloroplasts can actually divide and fill up inside of a larger cell. Okay, well, how did these mitochondria and chloroplasts get here? And there's actually the same answer might even be true for the nucleus. But the idea is generally called endosymbiosis. And this is the idea that a long time ago, before there were eukaryotic cells, essentially, maybe a bigger cell, a predatory cell, engulfed an aerobic bacterium, and maybe later on engulfed a cyanobacterium, which is a photosynthetic bacterium, or maybe vice versa, I don't know. Those cells, instead of being digested, that host cell kept them alive and allowed them to grow and multiply. Inside of the cell, it offered some protection, but it also gained some function, some more efficient metabolic strategies like photosynthesis and aerobic cellular respiration. And then later on, after billions of years of evolution, those became the mitochondria and chloroplasts. I shouldn't say billions, maybe hundreds of millions, or maybe up to a billion. So, so that's maybe where these modern eukaryotic cells came from, this endosymbiotic origin. Cool. Okay. So that was our first big grouping of structures, the nucleus, the mitochondria, and chloroplasts, just because double membranes, own DNA, potential endosymbiotic origin. All right, the next group of organelles is the endomembrane system. And endo means within, membrane just means membranes, right? So this is a system of membranes inside of the cell that all work together. And it's composed of, well, actually the nucleus is part of it. So that's kind of the control center, right? And if we look at the outer layer of the nuclear envelope, that is actually continuous with the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So let's break down those words, rough and smooth. Hopefully you know what those mean. Endo means within, right? Endoplasmic really means within the cytoplasm, and reticulum just means network. So, so there's a network of membranes inside the cytoplasm. Some of them are rough, some of them are smooth. Okay, why are the rough ones rough? Well, because it has all these little dots on it. When we look with an electron microscope and we take a closer look, we figure out what proteins are in those dots, and it's actually a mix of proteins and RNA that forms ribosomes. And ribosomes are protein factories or machines that build proteins. So, so essentially this rough endoplasmic reticulum is responsible for building proteins that are either going to be enclosed in a membrane or they're going to be embedded within a membrane inside that phospholipid bilayer. So any membrane associated protein is going to be built by the rough endoplasmic reticulum. Oh, okay. So for example, we have this protein for export that's labeled in green here. That's going to get built on the in the rough ER, and it's going to pinch off into a little transport bubble called a vesicle. And then that is going to go to the Golgi apparatus. These vesicles are just little transport containers. So you could think of them as like maybe the shipping containers, right, that you could put on a truck or you could put on a ship. And then the transport vesicle takes it to the Golgi apparatus, and there it fuses with the cis face, cis meaning near, right? And trans meaning across. So, and this is either near the nucleus or near the rough ER and across from it. And then that protein is going to be modified and sorted and packaged. So you can see some little modifications happening here as it goes through these stacks. And I like to think of the Golgi apparatus, this structure here. It's kind of like a stack of pita bread, pocket bread, right, with the spaces inside. And as it goes from one stack to the other, actually it's the same stack, it just kind of matures and gets new, there's new arrivals, new processes that happen along the way. And then eventually it reaches the transface, and that can pinch off to form another transport vesicle, and that can go to the outside of the cell if that vesicle fuses with the cell membrane, or the plasma membrane, I should say. Okay, so that is kind of cool. It's this whole system. The nucleus has the instructions reach a ribosome, a protein gets made, that protein gets transported from the rough ER to the Golgi through these vesicles, and then it gets modified, and another vesicle can take it to where its destination is, right? Okay, what about the smooth ER? Ooh, the smooth ER has a lot of different functions, and it's actually responsible for synthesizing a lot of the membranes. It's involved in metabolism of fatty acids. It's synthesizes steroid hormones like estrogen. What's the other one? Storage of calcium ions, especially in our muscle cells. There's a lot of smooth ER in our muscles because of the calcium storage. Detoxifying chemicals like in the liver. The liver has a lot of smooth ER, which is where alcohol gets detoxified. So there's a lot of different functions for the smooth ER. But it doesn't make proteins because it doesn't have ribosomes. Okay, the last member of this endomembrane system are the lysosomes, which aren't really pictured there on the next slide. Okay, lysosomes are little compartments that are contains proteins, and it's usually a very, again, if it's very acidic or very basic pH levels. There's a lot of digestive enzymes inside. It would be very, very bad for a cell to just open up a lysosome while it's inside the cell. So what lysosomes do, if let's say that's a cell that engulfs food particles or bacteria, a lysosome, that gets engulfed, and the lysosome can meet up with that and combine its digestive enzymes, and it just breaks down its food into the component the component monomers. So it breaks down the proteins and polysaccharides and lipids, breaks them down into smaller bits that the cell can actually use. So lysosomes are kind of like the recycling center, where the big things that make up cells, or maybe big things that are other cells that a cell feeds on, those get broken down into usable parts, and then waste can be excreted. Okay, all right, so there's our endomembrane system. All right. Some of the other cell structures, there are these organelles called peroxisomes, and these perform very specialized chemical reactions, in particular breakdown of fatty acids, which actually produces some really toxic byproducts. And so the peroxisomes can contain those, and then they also make hydrogen peroxide in the process, and that can be detoxified with a special enzyme called catalase. And we haven't talked about enzymes yet. So again, this is kind of foreshadowing the next module. But that is what peroxisomes do. So, okay. Okay, ribosomes. So we've actually already talked a little bit about ribosomes in the context of the rough ER, but there are also some free-floating ribosomes that are just in the cytoplasm. And the ribosomes are, I don't consider these to be organelles because they don't have a membrane around them. The ones on the rough ER, they're organelle-associated ribosomes maybe, but the free-floating ones, they're just kind of little parts, right? Some people will call them organelles, but then they have to kind of change the definition of organelle to make that work. So what ribosomes do is they build proteins. So they take the instructions that came from the nucleus, this RNA, which was kind of copied from the DNA, and that meets up with a ribosome, and then a different type of RNA uses that information to put a particular amino acid in a particular spot on the chain of amino acids that is going to become a protein. Ooh, this kind of links back to those macromolecules, right? So ribosomes are really, really important for the cell. And all cells have to have them because all cells need proteins to do the work of that cell. So, okay, what else have we got? Here we are kind of getting into the non-organelle parts of the cell. There's the cytoskeleton, which is the scaffolding of the cell, which we saw on that first slide. Here we have even fancier molecular stains to highlight parts of the cytoskeleton. I think the green is the microtubules of the cytoskeleton, and these are like the largest structures. And these are just big hollow tubes made up of proteins called tubulin, if you need to know. And these tubulin proteins can be stacked together, or they can be broken apart if the cell needs to move or change shape at some point. So another type of protein in the cytoskeleton is highlighted in red here. And these are called microfilaments, or sometimes called actin filaments. And you have a lot of these in your muscle cells that interact with motor proteins to cause those muscle cells to contract. And then there is another type of cell that are called, another type of filament called intermediate filaments that are kind of intermediate in size between the microfilaments and microtubules. They're made of keratin, and this is kind of a longer lasting part of the cytoskeleton. And there's a lot of this in the nuclear, inside the nucleus. There's a lot of these intermediate filaments, and they provide kind of it's somewhat flexible, but also somewhat permanent arrangement for the cytoskeleton. Okay, why are we talking about the cytoskeleton? Well, if a cell needs to move, it's going to involve the cytoskeleton. So a lot of things are flexible within the cell. The plasma membrane is kind of fluid, but the cytoskeleton gives it support and also allows things to move in relation to each other. So like on the inside of the plasma membrane, we have filaments of the cytoskeleton. We can have motor proteins that transport these vesicles along microtubules. So like these little highways and little trucks can move along those highways. And we have those muscle cell contractions over here. When cells divide, it's the cytoskeleton that physically separates the DNA and then also physically separates the cytoplasm of those daughter cells. Movement of organelles inside of a cell, that's happening involving the cytoskeleton as well. So there's a lot of different, very important things that the cytoskeleton allows cells to do. Or some cells that move kind of like amoebas that kind of force their cytoplasm in different directions. That happens because of changes in the cytoskeleton and motor proteins. So, okay, a couple other cell structures. If we look at the outside of the cell, sometimes we see a lot of hair-like structures. Those are called cilia. Cilia is Latin for hair, I think, or maybe short hair. And those, my analogy, if those old Roman ships with plenty of people rowing all in unison, that's how cilia operate. They kind of, these little short hairs that wave in unison, and they can propel the whole cell through its environment, or they can be used to move materials to a particular spot on that cell. This is a paramecium right here, I believe, and it's got cilia that can move food particles to its mouth, and then it can engulf those and have a lysosome digest them. In human cells, the lining of our trachea is full of cilia to move mucus from our lungs out, right? And also the lining of the oviduct between the ovaries and the uterus, that is also lined with cilia to move eggs from the ovaries to the uterus. Okay, there's other types of cell structures that allow movement. These are called, well, a singular would be called a flagellum. Plural would be flagella. Most of the time we only see one flagellum, although some protists might have a few flagella on a single cell. So here we have sperm cells, and there's this tiny little head that is most of the cytoplasm and the DNA, right? Then this very long tail that kind of whips around, and actually flagellum is Latin for whip. And what this is more like is maybe like the propeller of a ship, or maybe like a submarine, right? It's got just a single big propeller that moves it through the surrounding medium. Okay. Well, what else have we got? Well, I think that's about it. Those are all the structures I'm going to talk about. This lecture video is getting kind of long, so I will leave it here. I will say there's a really neat video. I really want to click on it and show you some of the cool stuff that happens inside the cell. But I don't want to make this video any longer. So instead, I'm going to put this link right below in our course materials, or in our course content module. So if you want to take a look at this, you can do that. And I'm probably going to show this during the lecture activity as well. So later.